Good evening, ladies and gentlemen. We now start with the last session for the day, Introduction and Overview of Bioinformatics. I request our keynote, Dr. Pawan K. Dhar, to please come on the dais. Professor and Dean, School of Biotechnology, JNU.
So I'd like to thank organizers for having me here today, and it's a wonderful experience to see young people in the audience who have stayed so long to hear science. And I congratulate you for your patience, and I hope that the rest of the day would be enjoyable as well. So my intent of today's talk is not to take you too deep into science. Of course, we will discuss science, but to combine innovation with science at the level of biology and try to impress upon you that when you do research in biology, there is a lot more than biology itself. You involve other disciplines. So at this age, when you're growing intellectually, it would be nice that you open up yourself to different disciplines, and then you will see at some point it will all integrate together based on the way your career moves in future. So all the research that we do is based on innovation. Without innovation, of course, you cannot move forward. What it means is that whatever existing database of knowledge is out there, you try to break that barrier and move ahead with the hope to generate new knowledge. So there was a survey of 1,500 CEOs across the globe some time back, and they were asked, what is the most important factor for the success of the future leaders? Amazingly, all of them in one voice said that it is creativity. They did not say sincerity, they did not say hard work, they did not say ethics, they did not say commitment, because this is a background constant. One has to have all these in order to be successful. However, creativity is a matter of choice for a lot of us. If we fall into the pattern of following what has been already done, you can be pretty successful, but the rise will be to a certain level. Now, if you want to really break the boundaries and rise in the career, then one has got to be creative all the time. Now, creativity is not what I'm discussing here today, but I just want to convey this message that whether it is biology, informatics, engineering, or any other branch of science, creativity is the foundation of everything. So let us run through one small example, just one slide, before we come to the main course of today's talk. Since there are many young people in the audience, I would like to ask you a question, and this is an interactive session. It is not a monologue, it's a dialogue. And so I'd be really appreciating if you interrupt me, something that you did not understand or you don't agree, please raise your hand and we can, stop it there. So I would like to ask this question to the young people in the audience. What is the color of the apple? If anyone can volunteer to answer this. Red color? Yes? So, if you take a static picture of this, what is on the screen, and I'm not talking about this faint black or faint white patches, I'm talking about the majority of the color of the apple. Okay. Obviously, anyone can say it's red color. It's the right answer. I'm talking the static picture, not of the actual apple, the way it grows over time, the color changes. But the static picture on the screen. However, even this data, which looks very simple, the question looks absolutely simple, the answer is not so simple. I'm just trying to convey the message of creativity here. You see, why did you say red color? Because physics taught you that whatever is reflected from a surface is the color of the surface. That's what physics taught us, right? But what it didn't tell us is that whatever is reflected is rejected. It is not accepted. Physics doesn't tell us that. You need to go beyond a certain level of physics. Whatever is reflected is rejected by apple, which means the true color of apple would be opposite of red. So what is that color? You have three primary colors, yeah? So red is rejected, and now when you combine these two, the point is the real color of the apple green? The red apple is the real color green. Likewise, you can do the same creative exercise with all the other colors, and you will see what is black is white, what is white is black. Now, we don't need to confuse ourselves because, beyond a point, this analogy is not very useful because when you're driving near the traffic light, if it is red, you'll say, no, this is green. So this analogy is okay in the classroom, or in a setting like this. But what I'm trying to convey is that it's all about creativity, and creativity need not be complex. It can be very simple and enjoyable. The foundation of all the science has been what we call as reductionism. Reductionism means that you have a complex system. Now, you are trying to understand the system, you are trying to break down the complexity of the system to understand what is it made of. So you say that this system is made of, this whole organism is made of longer or bigger systems. This is just one level of description. Scientists said, well, this is fine, but this is not complete, so we need to go deeper. When they went deeper into biology, they say, well, now you have a warehouse, you have a lot of organs. Theoretically, if you understand the function of each organ and you combine them, you have understood the whole organism. That was the foundation of anatomy and physiology. So now, when you look at each organ, the input-output of each organ, then you say, well, this organ itself is an ecosystem because there are so many different kinds of cells. Can I understand the organ just by looking at the input-output values? No, you need to go deeper. So people went to the level of cells. When you look at the cell, it looks very simple under the microscope. However, when you go down into the details of the cell, you notice that there are millions of molecules. DNA, RNA, protein, carbohydrates, lipids, you know it better than I do. One of the principal component of the cell that has been appreciated for many, many decades is the DNA or the chromosome. When DNA combines with proteins, it coils 7,000 to 10,000 times, it makes a chromosome. Okay? So it's the same DNA. It's like a thread. It is coiling thousands of times and making a chromosome. And the chromosome uncoils, it becomes the DNA thread. So DNA initially was recognized as the director of nuclear affairs because people thought that DNA is one molecule that is so important, it does everything. Because there is no appreciation of the other molecular role of other components in the cell. So DNA was everything, so it was behaving like a director. It took several decades for people to understand that it is not the director, it's actually a database of nuclear acids. It's just a database. Because this information, which is in a zip form in the DNA, it gets unzipped in the form of RNA molecules. And then many, many RNA molecules and protein molecules are made from one piece of DNA. So the information is zipped into DNA. DNA is okay, but the real deal is when you study RNA and protein, how they talk to each other. So the molecular level At this level, it is fine. However, now when you study the chromosome in its great detail, and you see that as the technology evolves, biology also evolves. When you had no microscope, Mendel was okay. He could give you that ratio and you're okay with this. But those ratios are from phenotype to genotype, genotype to phenotype. This is zoom in, zoom out, zoom in, zoom out. There's nothing in between simply because that was not the purpose of these laws of inheritance. The laws of inheritance were designed for something else. And everywhere, in a very subtle way, mathematics was used. Laws of inheritance, as you know, you do addition and division and obtain the laws of inheritance. So mathematics is used in a very, very preliminary way. However, as the more and more data comes in, you see that the description of the system changes with time. So what is this DNA made of? DNA is made of molecules, right? You started with biology. You saw an organism. You started with biology. And now you landed up with chemistry. Atoms are talking to each other. Chemistry is born. But atoms themselves need to be appreciated. So the next level will be that of the atomic level. Now, when you go and study an atom, what do you see? You see that 99% is space, rather 99.9% is space. An electron is just a probable pathway somewhere around the nucleus. The trouble is you cannot study space with microscope. So far, microscope has helped you up to the level of chemistry, and now it's a different ball game altogether. How do you study the space? And when you zoom out completely and you see the scan of the entire universe, you see that this is 96.5% space. Stars, planets, all the galaxies, they're just making 4.5%. Bulk is space outside, bulk is space inside. However, we still use the expression in the social interaction, I need more space. We already have 99.9% space. How much space do we need? Well, the atom, now from biology, you're landing up with the fundamental physics. We also left chemistry. Where did you lose biology on the way? Because this is completely physics now. There's nothing biology here. So since we are talking in the biological setting, it is better to zoom out from atoms. Because what are atoms? They are nothing but bubbles. This is bubble of space. So the DNA layer looks quite comfortable for us. So we would like to study DNA not because it's an important molecule, but also because it is the molecule within our space of resolution. So when people studied DNA, what did they find? There was a big human genome project, right? Ran for across several continents and for many, many years, and said that around 24,000 was the initial number. It has started decreasing to close to 22,000, 21,000. It's anybody's guess. What did it reveal? It revealed that 50% of our genes are exactly what you have in banana, from human to banana. Exact copies of the same genes. 50% of our genes match worm, called C. elegans, on which somebody got a Nobel Prize. So in other words, the Human Genome Project told us we are half banana, half worm. Well, this is good, humbling experience, nothing wrong with that. But it also tells us that nature did not spend her time in inventing parts. She spent her time in rewiring existing parts and generating complexity. This is intelligence. Well, that is what you get when you start combining these parts together. So, DNA layer, because I was told that there are many people from industry here and also a lot of students, so I thought, let me decorate the DNA a little bit, which will look comfortable for the industry. So, what are the applications? At the level of DNA. Well, if you ask me, the molecular diagnostic market is just exploding. And India has just 1% of the global market, just 1%. There is huge scope. And, well, there are different surveys with different numbers and so on. However, it's a big market out there. And there are startups looking for investors. And if you're a CEO of a company or you're driven by industry, then it's a small investment and the returns are realized in much shorter time scale compared to the typical biotech investments wherein the incubation time has to be at least 10 years. So that is the value of biology at the level of DNA. Not only the DNA, but also the protein, the RNA level. As more and more markers are discovered, you do faster diagnosis, you do better diagnosis, and also you advise how a certain disease should be treated in a specific person or a specific group of people. So you have personalized medicine. And this is coming as a combination of biology and informatics. Now you have a database of all the genome variations, and then you match it against that data, and then you predict what will happen to the person 5 years down the line or 10 years down the line. Well, so far we have studied what happens when you reduce the system. Okay? You split a system in terms of its components and you reduce it. What will happen? We have just gone through that. Now when you have a lot of parts, you have 20,000 genes, you have 100,000 proteins, what do you do with that? Of course, you cannot memorize them by their name or by their association with certain compartments in the cell. So you transfer all the data to the computer. You make a database of parts and database of interactions. So you transfer this. This is called modeling. When you're representing biological data in a certain form in the computer, we call it modeling. And the other approach of doing biology is if you have to think like an engineer. Can I construct an organism? Can I construct a bacteria from its scratch? So there are 3 approaches which have been predominantly used. The reductionism, which we have just seen, and integration, and the construction. These 3 major approaches in science. The first one we popularly known as molecular biology and biochemistry. These are the popular terms given for the philosophy behind these approaches. The other approach is more commonly known as systems biology. We're not talking of bioinformatics of a component, we're talking of bioinformatics of the entire network or cell-cell interaction. This is called systems biology. And the third, which has come in existence last 10 years, is synthetic biology. So I want you to appreciate that it's all about doing science. Whether you reduce the system, you construct the system, or you represent the system. And you do not know which approach will work where. And synthetic biology is more as an engineer approach to science. So we have seen how reductionism works. Let's spend the next 5 minutes in systems biology and see if there are any take-homes from there. If you think of weaving the data together, you think of integrating the data together, obviously your data comes at different levels. It is not just sequence. Sequence is a static representation of data. You have structure to negotiate, structure of the protein and RNA molecules. You have expression, and then you have interaction level, at least these 4 levels. So obviously, you need some tools to integrate the data, and that's where the computer comes in. That's where mathematics comes in, because you have to integrate based on certain rules. So there are certain steps in modeling biological systems. First, you make the list of the parts that is in front of you. And this part list is more commonly called as a database of biological data. Then you decorate these parts. Decoration means you describe these parts. You say, how are these parts looking like? In which organism? How do they express themselves? What is the range of expression? Do they fluctuate over time? We were talking the fluctuation of the color. What is the error bar? Are there any data conflicts? Suppose in your lab, in my lab, are we getting the same data? Are we getting 2 different data? If we're getting 2 different data, then whose data should be trusted? So there are many issues which come into this picture. So you need to be a good biologist to resolve it. Then you make a map out of the interaction. Once you have an annotated set of parts, the third step is to make a map. A static map, it gives a road map, but a static one, it does not move. This map does not change over the x-axis of time. Now, in order to make a movie out of the map, you do a bit of numerology here. You add numbers. So much of DNA in a cell, so many RNA molecules being made per second, so many protein molecules being made per second. Now you start qualifying these components with numbers, and you create a quantitative representation instead of purely qualitative representation. When you make a movie, we call it simulation. Simulation is nothing but you're changing the state of the system over a period of time by looking at the concentration of molecule as a parameter. So the concentration fluctuates over time. This gives a dynamic nature to the system. Like you have seen those people who make animation on the paper, when you flip the papers very rapidly, you will see some movement is generated. It's just like that. So then you use some analysis tools. When I was heading a team in Singapore, we had made this tool called Cellware. And at that time, there were 110 or 115 global platforms made by different people across the world. And we were fortunate to be classified as the top 10 by Nature Biotech paper. However, we can talk of science some other time. It is just that I wanted to indicate that the part information is not just enough, because the part information will give you a partial information, a partial snapshot of the system, which may or may not be accurate. When you weave this together, then you create a certain representation of the system. Now it starts becoming a real organism-like. So moving from science to applications, the current state of the heart, as I would call it in the pharma industry, a lot of medicines were withdrawn from the US market due to serious drug safety concerns. And they came to know about this much later. After marketing the drugs, people come to know that there is a problem with the drugs. And traditional methods of drug discovery are not helping much. How many drugs have people discovered for the last, let's say, 4 or 5 decades? Or are we combining existing drugs in different combinations to generate the response? So there needs to be a new way of looking at the drug discovery, and that's where systems biology comes into picture. How can systems biology help? It can help in the discovery of the drugs, the shortening of the drug discovery cycle, because the experiment that you used to do, let's say in the cells or animals, people are now trying to do the same experiment in the computer. So let's say you have a microbe, a harmful bacteria. You convert a virtual model of that bacteria in the computer and you test your drugs in the computer. Before you go for the other animal trials, at least it will not only speed up the whole process, but also it will narrow down your experimental space. So you don't have to do 1,000 experiments, maybe you have to do 100 experiments, because computational models give you that lead as to which experiments would be most useful to find out new targets. You can, in the computer itself, you can predict that if a certain drug molecule interacts with the cell, what kind of side effects will be created? This can be predicted. And so it's a combination of experimental data with the computational model. However, if you take it as an investment exercise, one needs to have really a lot of incubation time. It's more of an academic exercise at the moment. Of course, people in US are investing a lot in this area. But eventually one has to wait for many, many years, because you are building life from scratch in the computer. So you better be accurate. One can also predict toxicity. People are working towards building virtual cells virtual Mycoplasma genitalium. This is a pathogenic microbe. Virtual cyanobacteria for energy, bioenergy applications. People are working towards that. Virtual red blood cells, and this is the group that I joined in Japan. I learned systems biology from there. Can we construct, and recently we have constructed this virtual RBC model in our lab in JNU. And we have modeled certain hemoglobinopathies also. Virtual skin, it's a big payoff, 5 billion market. If you can model the hair growth, hair color, aging, so the color of the skin or the color of the hair and the growth of the hair, this can come together in a virtual skin project. There are many companies in Europe that are working towards that. So there are many opportunities one can think at the interface of biology and informatics. Virtual brain is a mega project in the US and Europe, and right now people don't have computational power to see this virtual brain. There are billions of neurons, billions of connectivity between neurons, and billions of molecules within each neuron. It is such a huge parameter space that you cannot run the model with the current, even the most advanced supercomputers, you cannot run the model. There is so much of information. So what they're saying is that maybe 10 years down the line, when the computational power will be 10,000 times or 100,000 times more, we can see brain as a slow motion movie, the whole brain in the computer. There is so much of data out there. And the payoff has been in an unexpected way. IBM has used, Dharmendra Moda from IBM, he used the current brain data to create intelligent chips to be installed in robots. Well, many years ago, I came up with this thought paper, is it time for a periodic table in biology? You know, after all, what is a periodic table? A finite set of elements, when arranged in a certain way, they can help you predict many things. Can we do the same thing? And if at all, what should qualify as an element of the bioperiodic table? It should not be infinite. That's very important. It should be finite, and it should be a funnel between different information layers. So we felt that this qualifies. However, there is scarcity of data, so we are not able to construct. Even if theoretically there has been some thought experiments, but still it is too early to construct this table, because the data that would link different layers is missing or is not complete. So now we have seen from reductionism and the modeling perspective, now the third layer, that is the engineering aspect, when you construct things. So the question is, can biologists think like engineers? When I say think like engineers, what am I trying to say? Can we construct a bacteria? Can we construct human cell? Construct, not study, but construct as an engineer. Why are we talking of that? It's because there are many similarities between engineering and biology. As you can see on the screen, a number of similarities, and that's what motivates you, that maybe what worked in engineering could as ballistic and unpredictable. This is an intelligence that has evolved last 3 billion years. So now if you're trying to kill a microbe, for example, and you say, oh, it's coming, a lot of resistant microbes are coming up, of course that will happen. Because these are autonomous systems, they're intelligent systems, and they're highly complex systems. You have to break the robustness at different levels. So to design well-behaved systems, one needs standards and rules of composition. This is what we learn from engineers. When you talk of engineering standards, they do not exist in biology simply because organisms use variation for their survival and evolution. They do not conform to certain standards of existence. So it is good to use engineering philosophy and instead of copying and pasting equations from engineering textbooks. It looks, as I'm talking, I'm losing audience, so probably I'll try to make it more interesting. So it's good to use engineering philosophy instead of engineering approach. Um, as I said, copying and pasting equations. Absolute engineering solutions do not exist for biology. Why? Because probably engineers never faced a problem as complex as biology. When I'm saying complex, you see, if you look at a Boeing 747 aeroplane, there are 7 million parts. There are hundreds of kilometers of wires. It's a gigantic system, right from the nanoscale to the macro scale. So wouldn't it qualify to be a complex system? No. The complexity here, what we are talking, is of a different level. We're talking of a cell which is so tiny that you can't even see this properly. We're talking of complexity as a function of probability, not as a function of numerical abundance. So, which means that the processes in the cell happen as probability. That means a transcription factor, this is a gene and this is a protein, it may or may not bind the gene. It may or may not bind. A lot of things can happen in between before a transcription factor binds a gene upstream to switch it on. Many things can happen, many things can go wrong. And whatever RNA molecules are made, maybe 90%, 95% will be destroyed by the time it makes protein. A lot of things happen on the way. Therefore, the complexity is in terms of uncertainty. However, we know that Heisenberg also made a career out of uncertainty. Why not we? So what's the solution? Solution is to, well, in the traditional setting, the system has been decomposed one part at a time, and we know this reductionism approach as molecular biology and biochemistry and so on. The new approach is to compose the system one part at a time. But composition means it's a big responsibility. You see, engineer has a much bigger responsibility than a physicist. Physicist can come up with an equation and enjoy the discovery and have a cup of coffee. But engineers need to use those equations to make a building. This is much bigger responsibility. So when you're composing biological systems, we need to make sure that they are controllable constructions. It does not go in a random evolution. So concepts like truth table, which were alien to biological setting, have started making appearance. And it is not a truth table that you see in engineering setting as 0 and 1. It is a quantitative truth table. Given a certain protein in the vicinity, what will be the state of the gene expression at a certain time level and so on. So synthetic biology, if you have to define in a very abbreviated way, is a rational design and controlled construction of biological components leading to applications. Applications, so maybe you can just have a look at this again, because I guess I have 10 minutes more, or? Okay, thank you. All right, so synthetic biology is a rational design and controlled construction of biological components. When I say components, I'm talking of genes as parts, and operon as devices, and pathways as circuits. So you have this abstraction from engineering: parts, devices, and circuits. This is all embedded in the term component. Now, Gene Core company is using bacteria to make rubber. This is commercial. This is not in the lab. They are synthesizing, making E. coli synthesize isoprene. In the normal setting, it would either not synthesize or synthesize very tiny amounts, if at all. But now, it is being engineered to make something that is used in making these Goodyear tires. OPEX Biotechnology is using yeast to convert sugar into bioacrylic. And bioacrylic, 80% of the greenhouse gases are reduced. It's less expensive. It's environmentally friendly. And the chemical industries, which are famous for polluting the environment, you completely eliminate that possibility. These are the commercial applications that are already in place. Okay. The long DNA synthesis that has been one of the frontier applications of synthetic biology. Earlier in a genetic engineering lab, if you have to construct some module, it will take you 1 year at least. Now with this technology, it takes you maximum 2 to 3 weeks. All you do is, you construct your vector in the computer, you email the sequence to the company and say that, I need a recombinant vector of a certain type. I have constructed everything in computer. I'm not going to do recombinant DNA technology. It's going to take me months and months of frustrating hard work. They will deliver you this recombinant vector in 2 to 3 weeks maximum. Okay. So earlier when the field started, the cost of synthesis was close to 80 rupees, 60 to 80 rupees. Now it is only few paisa. So that means people can now afford to accelerate innovations in biotechnology, which used to take a lot of time. Now people often complain, why does it take so much time to see the ROI in biotech? First, we cannot control this. Now we can of course control it. And second, if a human cell takes 24 hours to divide, and an E. coli takes on average half an hour to divide, you cannot accelerate this time span. You have to wait till it divides. What can you do? So that's why all this division, the natural process, it adds up to the time it takes to deliver an innovation in biotechnology. Well, we have a very simple mission in our lab of making our own genes. So this work was initiated when I was in Japan, and we asked why nature used a certain sequence to start the expression, to stop the expression. So it is the expression begins from here. Now we call it a gene. Expression begins from here, it stops here. What made nature to decide that? Did she try something upstream? Did she try something downstream? Did she try different variations of the gene? What made her think of this as an optimum sequence? And if she has not done all the experiments, can we do those experiments in the lab? That was the question we asked ourselves. You see, you have 3 types of DNAs. One is the one that makes protein. A bulk of it, 80% or 90% makes RNA. It does not make protein. So you have RNA coding genes. They do not make proteins. Now the definition of gene and the central dogma has changed. And you also have 10 to 15% of DNA that neither encodes protein nor RNA. It seems to be doing nothing as far as the expression is concerned. So we said, can we take the dark matter of the genome and can we make brand new genes of our own? So there's a lot of science behind that. I'm not coming to, because I know this is the end of the day and you need to rush back quickly. So I'm just giving the headlines. Maybe in another setting we can talk about the science. So we're making this encyclopedia of lab-made genes, and the payoff of this was very interesting. We have discovered new molecules against several harmful microbes, against diabetes, osteoporosis, Alzheimer's, cancer, malaria, leishmania, and so on. Where are these molecules coming from? The interesting thing is not from outside environment. A sequence that was considered junk in a yeast, we have experimental proof of converting this junk DNA of the yeast into anti-malarial molecule. Now we are patenting it. Likewise, a DNA sequence that was historically considered as junk in E. coli, we are reclaiming the junk and introducing a brand new drug discovery platform. So you define the gene based on bioinformatics, based on computational. These are high-end bioinformatics tools that you have to use, a series of them, a lot of filters into place so that you eliminate false positives. And it has taken us 5 years to reach this level of 500 molecule inventory that's ready to go for synthesis and patent. So user defines the gene and makes the gene and generates application. So our work has shown that the unused DNA, or the junk DNA, so to say, is a gold mine that nobody tapped before. It's a very simple process. Simple for us because we have been in this for the last 10 years probably. And, but the take-home lesson that we learned is that you own the IP of every molecule that you make. You do not make the molecule if the IP exists. Still, you are left with a large number of sequences. Remember, protein coding how much? In humans, 1.5%. 98.5% of human genome you can convert into your own intellectual property. Because these protein molecules do not exist in the nature, as far as the current knowledge goes. So we have worked, my PhD students have worked very hard, and we are hoping to offer something interesting for the community in the due course of time. You heard of genomics, proteomics, lipidomics, metabolomics, and so on, all the omics, because we are starting from junk, everything is a junk food, as many of us know. So my students like to call it as junkomics, as the new area of research. Well, we were fortunate, 2010, European Science Commission invited me for a talk, and then they officially declared this as a potential breakthrough. Then the National Academy of Sciences in the US, they were quite interested in this, and I was invited last year to, fortunately, to visit White House and share some of the experience. But the most interesting experience was the OPCW, Organization for the Prohibition of Chemical Weapons. And I was thinking, why did they invite me? Because you're talking to, it's like a parliament, and you have to talk to diplomats from 80 countries. And it's not a presentation, it's a dialogue, and you have to defend your work, and they ask you questions. So somebody asked me that now that you have made functional genes from what was called junk, can you make a lot of them? I said, yes, it's possible. And then the next question was, then, can you make bioweapon from your technology? I said, you gave me an idea, now give the funding. That was on a lighter note, but then we realized that we need to be very careful because it can go innocently in certain directions. So now we are matching it against the viral databases. Wherever any pathogenic organism is found, we match it against that, and then only we make it. It's absolutely safe to make it. And a lot of industrial applications, we are looking at new enzymes, new drug molecules, and so on. So we are looking for investment from the industry. These are bioinformatics driven and experimentally proven proof of the concept. It's a new drug discovery platform. We always face challenges, and we love challenges. And in fact, when the project started, my postdocs and PhD students in Japan, they refused to work on this. They said, there is no publication. Give us a project where there is a publication. Well, I said, this is an opportunity, but then everybody refused. So, of course, we respect their decision. And so we had to train a technician from Singapore. And then she did all the experiments, and it's very rare to have a lab technician as a co-author on a scientific paper, but that's what happened. And now, after that, it became a big story. So there have always been challenges, and this is my last slide. We receive motivation from an atom that says, be positive, even if you're always surrounded by negative things. So thank you very much for your kind attention.
I'm not a student like these kids. I'm a farmer. And how can we use it? Firstly, I doubt your data, because basmati is sometimes the qualities and other things are checked on the DNA basis. Mm-hmm. And the basic data I doubt, because of the human data which you said you have collected. And every human being is different than the next one.
Sure.
No one is the same.
Absolutely.
How could you have that data, and take it as a base? And then there was another thing. I went to Holland, and
yes,
they were testing some taste and things, basing on some people coming and they're testing. You could taste the same thing in a different way than I do.
Sure.
So we cannot rely on such data. That's the hang-up. And as far as the quality of produce we export or we use, and they need to be like on the basis of DNA, a scientist declares, and I know what is happening in the labs, you know much more than that. And we never get the true reports on that thing, and we suffer from it. There are so many questions, but I doubt the basic data. altogether?
So, thank you, sir. Thank you very much. It's a very important point. May I request you to kindly qualify this question by saying which data, because there were three type of areas that I was talking about.
You see,
Genomic data.
you too far off and far ahead of what I am into. I said about the farming, and I was talking about basmati.
Okay, okay.
Because this is, I never knew this, I would be attending such a class at this age. I'm 71.
It's an honor for me.
But can we have something
Yes.
like we need to test the plant like this, okay? The leaves, and we test it, and a quick process.
Yeah.
So that when it travels to the, you see, other country, anywhere in the world, and we are able to find the traceability. Because in organic is TT and TT, total traceability and total transparency. How could your this technology be helpful in this matter? Thank you.
Right, thank you very much. Well, that's a very important point that you have raised. First of all, the authenticity of the data and reliability of the data is that of your primary concern. You see, this comes up when you talk of testing drugs. Now, even after you test the drugs on a certain group of volunteers, and you do this for many, many years, still unexpected outcomes happen as the drugs were removed from the shelf, because there is so much of variation. There is so much of variation between our DNA that now people are realizing that in pathology labs, for example, you say my lipid levels are high or they're low. You cannot say the same thing about DNA, because there is so much of variation. So what is a reference DNA? Nobody knows about it. Yeah, please. Yeah.
So, you see, that's where I come to Ayurveda.
Yes.
Vat, Pitta and Kapha.
Yeah.
They have divided, and you, since you're Indian, you must know about it. And you look like Punjabi. So, if we analyze on that basis,
Yes,
I
sir.
found out that, you see, we had a habit. We were living Ayurveda. Like tarboos ke baad mein pani nahi pina.
Yeah, sure.
After the watermelon, you don't drink. After the melon, you don't drink. But after the melon, we were told when we were children that shakkar da pani pee lo.
Okay, okay.
And
okay.
see, you understand,
Yeah,
the
yeah,
disciplines in Ayurveda, Anupan, Anupat,
Yeah.
you see Vat, Pitta and Kapha, and then different constitutions.
Yes, yes.
But within the constitution, they have so much. Can we start thinking in those terms?
Yeah.
Because then we find the
Fantastic.
solution. Reason being,
Sure.
the medicines which were made and found thousands of years before,
Right. Sir.
they are the same and they work. Still they work. They haven't gone off the shelf or
Off the
whatever
shelf,
you may
yeah.
call it.
Sure.
But these drugs,
Yes, yes.
and you just said it yourself, and the public has to face it. And nobody, public faces the consequence, but not the one who's made it and sold it.
Yes,
So
yeah,
thi-
I
this
completely
is,
agree. I completely agree. Suppose you take a drug against so for killing a microbe, and this microbe changes its genotype or its DNA over time, and the same drug will not work after some time. You see, drug resistance has emerged in the community. Of course, that is the limitation, because you have limited knowledge. With a lot of efforts, you could just find one target, but microbe is more intelligent over time. So you have to again work. But I agree with your Ayurvedic approach, because I like to point out some work in that direction of your interest. And maybe Dr. Bhushan Patwardhan from Pune. So they have a lot. So now they are starting Genomics. So then you look at Vat, Pitta, Kapha, and then you see the DNA sequence properties of each group of individuals. This is being done in Pune University, and also there is a Mumbai group which is very active in that. I'm blanking on the names, but people are now appreciating that they need to bring science together with this. Yeah, yeah, of course, it's already a science. Yeah, yeah, yes, yes.
Whatever medicines or insecticide, pesticide, and I have here the photographs I can videos to show you.
Yes.
They affect the root. Then what we have evolved is we have started working on root instead of anything else.
Right, right. Yes, 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 yes. Absolutely, absolutely. Absolutely, I completely agree because the other the therapeutic approach that we are using, the Western medicine, so to say, is a short-term, acute, and it is not going to address the root of the problem. It's just going to suppress the symptoms for some time. Yeah, completely agree. So for the root cause, of course, you have to go for a fundamental classic approach. Yeah, completely agree. Yeah, yeah.
Hello, sir.
Anything from the yeah, please. Yeah.
I'm Pulkit. I'm pursuing my second year in biotechnology from APJ Satya University.
Okay.
And as you talked about the virtual brain thing,
Yeah.
as we know it, there is artificial intelligence, but
Yes.
it doesn't have an consciousness.
Yes.
So as you talked about that virtual brain, will it be possible to have consciousness in that virtual brain thing?
Thank you very much once again. Good question. See, we have not photographed soul. We have not, if at all, whatever the description are there. We have not studied the composition of life operating system. So we have to eliminate life to get the DNA. Okay, you understand that. We cannot get in the live situation. So essentially, in other words, you have to kill life to do life sciences, which looks very ironic, you know. So, but if you retain life, then the animal ethics groups are after you. That you're so you can neither retain the life nor well, life is, yeah. So, to answer your question, the brief answer is that I do not know the composition of soul, or I do not know what life is composed of beyond a biochemical description or the genetic description. Of course, there is another fundamental description at the level of intelligent energy or whatever you call it. And that's where the science is moving. You see, what used to be spiritual or philosophical, now it's becoming science. Harvard, some time back, started Harvard School of Divinity. Why? Now Stanford is into this, Berkeley is into this. Why? Because science has hit a limit. You say you come up with 10,000 genes, maybe if you have funding, you come up with 100,000 genes. How does it matter? Say please. Right. Yes, sir. Yes. The Sukshma and Karan Sharir, yeah. Absolutely. So Karan Sharir we do not know. Yeah, at this level of... Okay. Yes, yes. When there's only Karan, maybe... Yeah. Right, right. True, true. Yes, sir, yes, sir. Completely, yeah.
Interrupting.
Sure, 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 sure. Absolutely, absolutely. You see, time has come that liberal arts, what you call as liberal arts, that should be combined with the other, so to say, science in terms of the physics, chemistry, biology. And philosophy needs to come into picture. Because otherwise you just take a snapshot and you think that this is the complete picture. It is not. That's why these models that are made in the computer, they are just representation of reality at a very partial level. This is not reality. It's like if I place my hand here and the light is falling, you will see some shadow on this curtain. Now shadow is the model. What has happened to the real thing? We have lost a dimension. The moment you are studying shadow, which is a model, computer model, we lost a dimension. From 3D it became 2D. So 2D is not real.
So can they be reliable? That
sorry?
computational models, can they be reliable?
How reliable it is?
Yeah.
Well, it depends. Of course, that's a tougher one because if the data comes from different labs which is conflicting, then you usually go with the lab which is very famous. It does not mean that this is the true data. It means that it's the most convenient way of representing. But again, you need to go back and forth. So the first time you build a model and you predict something, usually the prediction is wrong. So you need to go back and forth, back and forth. It takes years and years together to build a model. However, it's not a complete model. It's very tough to make a complete model. So you only make a model for a certain question in mind. If your question is very well defined, you create a model out of that, and then you start experimenting on that model. But the accurate answer from that model depends on how accurate your data is. So it's like garbage in, garbage out. If the input data is not accurate, the answers will not be accurate. Yes,
Hello,
sir.
sir. I'm basically a software engineer.
Yeah.
You know, I have been working as a software engineer since last 10 years.
Mm-hmm.
And I studied data analytics as well.
Okay.
So you were talking about, you introduced data analytics in pharma. So, as a entrepreneurship, then what you can guide us, exactly where we should focus on,
Okay.
keeping the technology in India, the available technology in India.
Well, there are many areas of avenues of research. One is deep computing. I think deep computing and brain data, they are coming together. So if you are into deep computing, then you will see a lot of publications coming in that area, where there will be some underlying weak patterns that need to be uncovered with deep computing. The other one is the big data analytics. Big data, right now people are correlating with the traffic. The Google is using the big data tools, or the weather experts are using that. But now the big data tools are also going to be used, or currently being used, by the biological people, especially the brain group. So if you are looking at opportunities in terms of the application of computation or IT sector in biology, one is building new tools has been going on for the last 20 years in the area of bioinformatics. Now there are sufficient tools to understand DNA or proteins or RNA. This is a very highly specialized field. But then there are opportunities available to build virtual models of the cells. And that's where you need IT team and biology team together. As I said, big data is one opportunity, but a lot of it is not happening in India at the moment, unfortunately. So when you think of big data, you think of deep computing, you have to think of US immediately. The other opportunities are in bioinformatics services, wherein you, as a service model, you try to predict the effect of the drug on a model, or you build a model for the labs. But then there are not enough biotech companies to sustain that. And they are mostly going for the service model. I'm sorry.
Yeah, exactly. So the challenge in India, the challenge is that the technology which is available maybe in US may not be available in India.
Yeah, but there's one aspect which I missed, I'm sorry. There is something called as e-agriculture. E-agriculture, I think that is a fantastic application there. That's what Infosys started many years ago, and in TCS also, which means that you connect the soil chemistry with the yield of the field, with the market, with the government agencies. You put together as a mobile application. There's a huge unmet need around that. But you need to really tap into the funding agencies and right groups to get it. It's a fairly big challenge. Yeah. I'm sorry, sir. Yeah. Sir. Yes, sir. Mm-hmm. Yes, sir. Mm-hmm. Yeah. 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 Right, 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 right.
We have fought in India, negative and positive. And that's where we go into the conscious level, with the conscious and the physical, because
Yes, I completely agree. Yeah,
the
yeah.
spiritual science says
Mhm.
the receptivity here is 1,000 times than the receptivity in the physical level.
Right, right,
Like
right, right.
Einstein
Yeah.
says, I have only understood up to 15, I've only used my brain up to 15%. An intelligent person
Yeah.
uses 7.5%. And a person like me would use 6 to 6.5. And below that is almost a group carrier.
Mm, yeah. True, 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 true.
But you see the yogis in Himalayas,
Yeah.
so many yogis, you see they're here and beyond it.
Yes, 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 absolutely.
You can
Absolutely.
use this science and coordinate and cooperate with the, conscious science.
Yes, yes.
Lot of things will come out, lot of solutions will come because
I
understanding
agree.
level, the receptivity level is 1,000 times.
Absolutely, absolutely. As you subtract the data from your mind, basically somebody made this back of the envelope calculations that we have 60,000 thoughts a day. That means if you have 60,000 units of energy, that means one energy unit per thought on a theoretical basis. Now, if you reduce the number of thoughts, maybe 10 times and 100 times, you have more energy unit for each thought, and this is what is called intelligence. And that's why when our parents said that you concentrate, you focus, what they are saying is you reduce the energy, you reduce the thought budget, you will be automatically intelligent. It makes sense, yeah. Thank you very much.
So we'll just take
Sorry,
last
sorry.
question of the
Sir,
evening, please.
Kindly focus on stem cells.
Stem cells.
Is a miracle cells, we heard.
Yes, sir.
And many uncurable disease can cure by stem cells.
Yes, sir.
So
Yes, sir.
kindly some place on stem cells, sir.
Wonderful, thank you very much. Yeah, of course, stem cells are extremely important storehouse of all the information, and now the technology is such that you can, if you have the methods in the lab, you can convert blood into brain cells. You can convert blood into heart cells. This technology exists. So if you have to, for example, stem cells are so important that if you have to, let's say you have to test the drugs for Alzheimer's. Now, where do you get the models of Alzheimer's to test the drug? So what people are now doing is they are converting the skin cells of Alzheimer patient into the stem cells and converting that stem cell into the brain cell and testing the drug of the same patient. So the technology has gone very, very far away. But we really need to focus on India. So there are stem cell banking. Of course, it comes with strengths and limitations. But people are now trying to come up with this IPS bank for our Indian community. A lot of universities are now funding big programs in stem cell research. So I think this is one of the promising areas for sure. Thanks for highlighting that. Yeah, sir. Yeah. Oh. Yeah, I agree. A lot of things need to be
Yeah.
done. Yeah, thank you very much. Yeah.
Thank you so much, Dr. Pawan Dhar, for your contribution to Wellness India conference. Any other questions would be entertained offline, please, because as it is, we're running a little short of time. Doctor, please accept a small token on our behalf, a token of appreciation from our side. Thank you so much.